Hello, ladies and gentlemen. I welcome you for this fourth talk on this series. Today, we are going to talk about oncogenesis and normal cell differentiation and single oocyte and egg cell, leading to vast variety of different tissues. So, how does the genetic logic support it? How does it work? So, we'll talk about it. So, let me share my screen. Today's topic is oncogenesis, cell and tissue specialization. Of course, oncogenesis is a bad word. It means cheating on the cell intelligence and somehow hoodwinking it to produce wrong type of cells. And uh, of course, viruses do make the cells to generate the wrong kind of proteins as well. So, how can one cheat the cell intelligence? And of course, how does the normal intelligence of cell work for differentiation into various kind of tissues? Now, this is the fourth talk in this series of 12. To summarize whatever we have been discussing in the earlier videos, this is how complex a chromatid looks when it gets spilled out. That is, it gets unzipped and spread all over the DNA strand. Okay, and of course it is wound on histone bobbins which have to be shrunk to expose the double helix ladder, which has to be unzipped for reading, transcription and translation, and even for cell division it has to be fully unzipped. So this is what we were starting. Of course, there are a lot of intracellular security mechanisms, if you see, like for example, any mutation at a single strand of DNA. Suppose there is a single strand change in the DNA, one half of the DNA strand changes. Okay, it will fail to rezip after the process is over. It is supposed to rezip into assembled chromatid for cell division. So there it will fail. So hence at that point it can get detected if there is something wrong going on. So that is one likely safety mechanism which is there. Okay, and if there is such an offender which is failing to rezip the thing, it may get dissolved and until it gets resubstituted with the something that is right, where the zipping is permitted. Full zipping happens between both the halves of the DNA. Yeah. Second mechanism is codon and anticodon matching at rRNA to prevent framing error. Okay, because there are two molecular conveyors. One is presenting a string of codon, other is presenting the string of anticodon with which the tRNAs are dangling down. Okay, so you cannot miss by one pitch or two pitch. The framing error gets prevented, and also intrusion of a wrong mRNA in between can be prevented because its counterpart anticodon will not arrive. Okay. Third mechanism is that wrong protein getting synthesized. Even if wrong protein is somehow synthesized at rRNA, it would simply get clipped by the shape and size of birth canal that is available. That is the interspaces of the cytoskeleton which are available there. Okay. So specific interfaces in the local cytoskeleton will kill something that is not conforming to the shape and size that is permitted. Right. Fourth is cytoskeleton will detect the presence of unwanted debris, intrusion, odd entity in cytoplasm, and the same will get promptly lysed. Okay. And protein sensors floating on cell membrane will block entry of any unidentified macromolecule into the cell. So these are the different levels of protection which are there. And to top it, when the cell detects overwhelming malfunction, it could commit suicide. That is, it could suicidally rupture the lysozyme scissor, and whereby the entire cell can get dissolved. Okay, so that is the ultimate. If nothing else works, that cell can commit suicide. Okay, so these are the 6 levels of defense, that is security that against any likely corruption. And that brings us to some tricky questions which we can attempt to answer. That if it were possible to surgically replace nucleus of a cell, suppose we isolate the nucleus of a cell and plant it into another cell, would it change the way the cell would function and divide further? Okay. Practically it is possible to do or not is besides the point. The logic is that interspaces of cytoskeleton may not permit something that is not a legitimate entity into that ecosystem. Okay, so that is the likely answer. And in most mythologies, we have the concept of half human, half elephant, half fish, half lion, etc. Okay, nursing avatar and stuff like that. Merlion in Singapore, for example. Yeah. Uh, Surely people may have attempted cross fertilization between animal species, but obviously it has not worked out at genetic level graft. Although in simpler plant kingdom, it is possible to do a grafting at the stem and can have some hybrid species grown like that. But through genetics, it is not known to work. Okay. Another tricky question is that okay, the lizards are known to shed the severe tail and they can regrow the severe tail. Okay. But can the severe tail, if it is nurtured into a system, can it grow into a new lizard? That is the question. So again, the direction in which the cell division will be directed by the genetics, whether it is possible to reverse and from a cut tail you are from a hair, can you grow the full human being, for example? Okay. So the question is something like that, but obviously it doesn't work like that. Okay. Next question is, are there any DNAs outside nucleus? Yes. Mitochondria are known to have the DNA, loose DNA strand, which control their growth and multiplication autonomically. And the same is not controlled by the nucleus. It is autonomous in cytoplasm. Depending on energy requirement, they multiply and orient themselves at the location where they are required. Okay. So, yes, there are DNA strands which are there outside nucleus in cytoplasm. Okay. And mRNAs made from DNA in cytoplasm may be reaching out to some rRNA which is there in the proximity, and they could be making some protein and vice versa can also happen. Probably that is the reverse translation can also probably happen, that from mRNA you can work back and get a DNA, right? From a protein you can work back and get a corresponding DNA planted. Yeah, probably it might be working both ways also. And DNA in cytoplasm could randomly get substituted on chromatids because whenever there is a big movement of spindles which are doing cell division, okay, so and at that time the chromatids are fully unzipped because they have to replicate and develop the other half. So at that time, it is just possible that complete DNA strand, that is both the halves of the strand, can get substituted. That complete DNA section can get probably substituted on some chromatid and it can go in the nucleus of daughter cell. Yeah. Now that is the premise that is leading to oncogenesis and cells producing wrong type of daughter cells, so on. Yeah. That's the only way it is possible because unless it goes into nucleus and goes to daughter cells, okay, it would not happen from cytoplasm directly. Oncogenesis is the phenomena of cells multiplying wrongfully. That is, they are cheating the intelligence of the and the defense mechanisms present in the cell. So oncogenesis, same as carcinogenesis, it means uncontrolled wrong type of cells getting produced. Okay. They consume the material and energy resources in the system, which is meant to be highly optimized otherwise. So wrong type of cells, they may not do any harm expressly, like they may not be something like poison, but then they do consume lot of energy, lot of resources, which are otherwise optimized in biological systems. They are rationed. So often, and their physical presence and uncontrolled growth may lead to some physical obstruction, physical crowding, cluttering, etc. Some things coming under pressure, some things growing uncontrollably, etc. And eventually, it may impair, most of the time mechanically, the system's functioning. So the vital systems of the body, that is circulation system, breathing system, etc., they can get impaired with all these things. And body defenses can get overloaded fighting and trying to destroy them, because sooner than later they will be identified as offenders. So the body defense will go all out trying to destroy them, but it will have to have a lot of overheads. Will have to spend a lot of overheads in trying to fight and destroy them. Okay, cancers are nasty because of simple reason that the problem surfaces few generations down the line from where the root cause begins. So genetically, the root cause may have begun 2 or 3 generations of cell earlier. Okay, and somewhere down the line, okay, that is precursor cell is somewhere way up in the ladder, and after 2 3 levels down, after 2 3 daughter cell generations down, then the problem starts surfacing that there is something wrong. And there could be a defective genetic archive having been passed through ancestral legacy also. So in which case, right from egg cell, everything has the genetic database corrupted. Okay, so that can be a widespread cause of oncogenesis at several places. But most of the time, it is found that it is there in one particular path of cell differentiation. Logically, it is impossible to correct the root cause trail in retrospective. You can't go back 3 generations and correct and re-execute the code correctly. Okay, you cannot reverse and re-execute to cure the problem. So the problem has already happened, and now one has to live with it. So that's the problem. Mostly, only the problem growths are, I mean, the way they are being treated, they are thought to be surgically removed, or the wrong cells may be selectively killed through chemotherapy or irradiation to control further proliferation and damage. Okay, so this is usually the approach that is there. Sometimes the precursor cells, one level back, like bone marrow transplant is attempted for blood cancer, etc. So sometimes such things may give result, but if it is several generations upstream, then it is difficult. And unless code corruption happens at chromatid, it may not get passed to daughter cell. That's what we said already. So something that is there in cytoplasm only, at best it can work like a virus and can synthesize some wrong protein in that cell. But it may not get passed on to the daughter cell so easily through genetics. Okay. So DNAs or RNAs present in cytoplasm may result in synthesis of wrong protein, but it would not result into cell division and wrongful differentiation of daughter cell. Okay. So let us see how oncogenesis is initiated. Some viruses will cheat the cell surface defense and forge entry into cytoplasm. That is, the sensors on the cell membrane will get cheated by the shape that is presented by the virus, okay, by the key that is, by the master key that is inserted in the lock, and the lock is opened and the entry is forged into cytoplasm. And then the virus may be carrying strand of DNA or mRNA, which could try to get substituted during cell division with certain segment of valid DNA. Okay. So some viruses may bring their own mRNA and start producing wrong type of proteins at rRNA, consuming the cell resources, energy, and disrupting the normal processes. But still, they are harming only the cell which they are directly attacking. Okay. And some cell divisions later, these mRNAs may get back annotated on DNA, and then they corrupt the genetic code, and then the wrong type of daughter cells may get wrongfully produced. So that is the root cause of oncogenesis. And carcinogens are the said to be the materials which defy the cell protection at cell membrane by mimicking the original key with master key, for example. And they are capable of opening some walls and inducing phagocytosis and increasing the possibility of viruses and some extraneous DNAs and mRNAs to sneak in. Okay. So generally, it is said that oncogenes are the ones which damage DNA, but damaging DNA is a very broad term, so to say. However, there is a silver lining in oncogenesis also. that cancer cells are generally much lesser robust and much lesser agile, so that they get killed relatively easily with irradiation or chemotherapy as compared to normal tissues. Although, yes, there would always be some collateral damage, certainly. And normal cells and then existing environment around them will reinforce mutually, while oncogenes produce products are not supported, nor do they support the local ecosystem. So they are odd man out. So the ecosystem does not support them, and they do not support the ecosystem either. So they can get identified very easily, and they can be isolated relatively easily. Okay. So that is what normally tends to happen. And usually the corrupted genetic code is restricted to some particular differentiation pathways, while the other pathways may still remain normal in the genetic activity. Okay. Some other uh, points which are affecting oncogenesis adversely, of course, that feedback and growth dynamics from other tissues will not reinforce the corrupted pathways. So after a while, the corrupted segment will run down and becomes lesser expressed and lesser effective, and it may even eventually get substituted or back annotated with the required code. There are some chances that logic may correct itself, okay, because it is not supported. And few generations down, the defective genetic code might even become extinct progressively. So what we once regarded as carcinogens at one time are no longer found to be carcinogenic now, because by now the cells have learned about their structure, and they have reinforced their defenses, and they are modified their sensors to discriminate and block them. Now that is also one phenomena which happens by way of positive evolution of intelligence. Talking of good things, normal things, not the abnormal things. Well, the cells do differentiate a lot into various kind of tissues, and the starting point is of course fertilization, where two haploid cells. This is a haploid cell division where you can see that the chromatid is getting split in the middle. Okay, and then the complementary things are built in both the daughter cells. This is the view of fertilization. This is a sperm penetrating an ova. You see, sperm is just like an arrow, it carries just about the nucleus, whereas ova carries many things. Okay. So, how does normal cell differentiation starting from oocyte takes place? The ova and sperms are haploid cell. They have 22 chromosomes, not pairs, just 22 chromosomes. Sperms just about carries the nucleus, as I said, but the seed cytoskeleton and food resources for the aggressive growth that should follow after fertilization has to be catered to by the ova. Okay, so mother cell contribute to the environment, the growth environment, where the cell division, cell multiplication takes place post fertilization. And of course, in mammals, the eggs are not laid outside, but in other animals, of course, the eggs are autonomous enough. So they are pre-packaged with the food and nutritional resources to support the growth into a full living organization, a living organism. But of course, in mammal, it still requires supporting system, uh, the blood flow, etc., from maternal side to cater to the requirement of logistics. And apart from prepacking the nutrition, ova is also specialized to latch onto the uterus wall for mammals, and it's able to source the resources for aggressive growth activity that follows fertilization. The main function of this cell is to multiply and transform into cytoblast, which is like a hollow circular ball, hollow spherical ball, which later develops into there is a septum in the middle, which develops into a three-layered embryonic sandwich. Okay, so the three layers are ectoderm, endoderm, and mesoderm. That's what it is called. Okay. So ectoderm forms the outer layer, endoderm forms the inner layer, that is the alimentary canal lining, and mesoderm forms most of the other tissues. So mesoderm comprise of mesenchymal cell, which differentiate into many differently specialized tissues and organs. Okay, so this is the embryonic sandwich. Ectoderm, endoderm, mesoderm is shown here, and these eventually they all endoderm diversifies into parathyroid, liver, pancreas, and all these glands, and also including the sex organs. And mesoderm, of course, bone, fat, cartilage, muscles, etc. And ectoderm is, of course, skin and nerves and sensors, etc., which are there. So these are the various different type of functional tissues that emanate from the egg cell, from one cell, fusion of two cells, two haploid cells, and the same genetic database is leading to such diversified properties of tissues that one gets. Okay, so a normal cell, of course, these are the stem cells or inner cells. This is a fusion of two cells, zygote. This is an embryonic sandwich. Then you take the stem cell, they can diversify into different. So what are those tissues? Of course, bones, cartilage, which form the rigid, flexible structure and kinematic pair joints. Okay, knee joints, arm joint, etc., which can magnify the movement through leverage and through joints. Then there are muscles which cause the movements and which apply force. Then there are cells that selectively absorb from one side and transport actively to the opposite side. Consider the intestinal lining cell, for example. They absorb the foodstuff, the digested food, selectively and transport it to the blood vessels on the opposite side. Okay. So obviously their cell specialization is modified into a transportation and selection agent. Okay. Picking intelligently and transporting it to the cell across. Okay. So yeah, and then of course there are cells which move with the blood. So there are surveillance and carrier cells that carry oxygen, CO2, and resources, and of course the immunological information, etc., for protection. Then there are cells which are primarily meant to secrete, and they make all kinds of glands, several glands that are there. In this talk, we are not going about describing the organs particularly, nor are we going into details of all the glands, etc. We are, as a type, just discussing several different kind of distinct tissues and what specialities they would have, okay, in their functioning. So we are restricting in today's talk up to there. Okay. So cells that primarily secrete and make glands, then cells which form the various walls of various vessels. Okay. And then of course the external skin and the sensors in the skin, etc. And last but not the least are the intelligent communicating networking cells. Those are neurons. Okay. So these are the distinct tissues in which the same egg cell can diversify. Okay. So a living organization, a living person is like a living city, for example, where it is made of cells. So if you liken cell to human being, in a city you have doctors, engineers, lawyers, police. So like that, there are specializations in cells, and they assume a specific function in the overall cooperative system that sustains the higher form of life. Right. So that's what it is. Now there are multiple differentiation pathways starting from mesenchymal cell. I'll not go too much in detail. This is a borrowed picture just for visualization. Whatever is there with yellow highlight is my own thoughts. Rest is pictures just to make the viewers visualize, okay, what I'm trying to say. So usually oncogenesis, suppose some corruption happens here, this path will be only affected. Okay, the other paths may progress normally. Okay, so if during the growth, unless the egg cell itself is having a corrupted code to start with, then everything can be affected. Otherwise, usually by far we see that either you have a bone cancer or you have blood cancer or some specific cancer at somewhere. So one of the pathways is affected somewhere on the way. So it is restricted to that particular pathway. Now, talking of bones and cartilage, of course, the inside is the forming surface, outside is the dissolving surface, and it grows into the layers. Okay, so osteoblasts will exude matrix of protein fiber, which becomes kind of a fabric that binds calcium, and ossification takes place. The calcium and phosphorus is stored there. It is used as a storage as well as it forms the hard inorganic structure, giving the inorganic strength to the bone. And osteoclasts will dissolve away the bone from wherever they are not required, that is the outer surfaces, and the joint carving function wherever it is juxtaposed with the other side of the joint. Okay. So to match the shapes, to suit the shapes, yes, bone carving function is there. Cartilage is the softer version and it is a more flexible variant, formed in a similar way, and they form structure like external ears, this one, joints covering the joint capsule, that is the uh, soft bag in which the joints are, okay, and joint cushions and ligaments and whatnot. And bones are also storing calcium, phosphorus. They also synthesize bone marrow and interact with chemical messengers and hormones. The beauty is that all these hardwares in biology are multifunctional always. They have more than one function. Everything. You see. So bones are anisotropic in engineering sense, and they have tailored strength according to the requirement documented on genetic archive. Okay, so they are strong where they are required to be strong. They may not be so strong where they are not required to be strong, and they may lose some weight and they may become light where it is required to become light. For example, people living in mountainous terrain, they have lighter but stronger bones. They are known to have less body weight because they have to climb vertically on mountains and carry weight on their head. So they are very sturdy Sherpas, but their bones are lighter. Okay. So there are some such genetically programmed specialties. For example, if you see goldsmith community, most of them used to blow a lot in the to heat the gold and silver. So mostly they have chubby cheeks. So genetically they have got modified and they have chubby cheeks. And farmers will have big legs and hard palms and so on. So such genetic specializations are always there. Then muscles are there. Of course, they are telescopic actin myosin assemblies, which are somewhat like a hydraulic or pneumatic cylinder in engineering terms. So they can contract or they can be relaxed depending on the chemical messenger present, and which can be presented through the nerves that are coming there. Okay. And larger muscles will usually be attached to the bone through some collagen kind of fibers. Okay. And they form the kinematic pair, that is between the joint. It will be spanning this and this bone. So if it contracts, it will have a leverage advantage of the bone structure, and it will magnify the movement by physics. So there are smaller muscles, of course, which do eyeball movement, focusing of eyeball, focusing of eye lens, batting of eyelids, control of vocals, moving of tongue, operating of epiglottis, opening and closing of sphincter muscles, for example. Okay, so there are varieties in muscles, then they are omnipresent in the body. Heart muscles, of course, go in rhythmic cycling movement, affecting the pumping of blood. And muscles also surround alimentary canal. They have the peristaltic movement, and they also operate the diaphragm. They propel the food down. Okay. They could also apply gentle squeeze to the glands to release the juices. For example, they can squeeze the bladder and they can possibly alter the shape of blood vessels to alter the blood flow pattern in the vicinity of some infection or something. Yeah. So one interesting class of cells which are specialized are, of course, the cells that selectively absorb, ingest, and pass through the opposite side. These are the intestinal lining cell or alveoli in the lungs. Okay. So intestinal lining cells have vilification, that is the surface enlargement is there on the food lumen side. So they are in the contact with the food that is flowing down, and they selectively ingest the large food particles and let the smaller molecule diffuse through, probably. And the cytoskeleton spanning end to end. Usually the cytoskeleton in a typical cell is supposed to be almost radial, where it is doing phagocytosis, it is sucking something in a vacuole, and it is digesting it with lysozyme, okay, within the cell. Now, but this one is specialized where the cytoskeleton is spanning across from wall to wall. So it is ingesting from one wall and it is delivering on the opposite side wall. So that is the specialization. Instead of radial orientation, it takes pre-digested food and propels it across to the bloodstream side. Okay. So intelligence of identifying the food on one side and the active mechanism to pump the food or propel the food to the bloodstream. Both the functionalities are there. It's a diversification, it's a specialization typically. Similarly, in alveoli in lungs, they have extended interfacing surface to air side, and oxygen and carbon dioxide is transported across actively in absorbed state across blood and airs, the air side lumen. Yeah. Then there are, of course, mobile surveillance and carrier cells which flow in the blood. They identify the offenders and they dissolve them, they engulf them. So these are the cells that are carrying out the defenses of the body. So cells in the blood, they carry the absorbed oxygen and carbon dioxide through the blood circulation system to everywhere, all over the body. And cells which defend the body through blood circulation by identifying offender bacteria or particles and neutralize them by lysing them. And of course, the cells also contain immunological information carriers to identify the known offenders and how to neutralize them specifically. So all the information and intelligence is carried by these immunological cells. Then there are vast variety of cells which form walls, layered structure. Just picture is enough. I mean, I don't have to speak much. There are different type of multi-layer or single layer, cuboidal or squamous cells in various applications that you can see. Yeah. So it's just the variety that I want to emphasize. Then, of course, neuron is the communication and intelligence network. These are the typical star-shaped, widely networked cells which carry the ionic currents. There are rivers of ionic channels inside. Okay, and it has one output and several inputs. It's like several tributaries leading to a one big river. And then when the level exceeds somewhere in some dam, the dam gets open and on output side some water gets released. So something like that happens through various inputs. Ions flow, and wherever the ionic level at the threshold exceeds, the gates are opened and a burst of ionic flow is released on the output side. It's on the axon. So they carry out the function of electronic networking and electronic logic realization, the sequence realization, sequential and combinational logic. So they are electrically insulated output trails which form the nerves. Bundles of outputs of neurons are the nerves, and they carry the electrical release of neurotransmitters which can operate the muscles, etc. So they are connected to all sensors and they also take electrical input from transduction. The various sensors are connected to neurons, and network neuron form the higher level of intelligence and physiological level control system of the body as a macro system. Then this is an example of the taste sensor. Similarly, there are smell sensors. They are something like extension of sensors which are there on cell wall surface. That is, when a shape will come and fit exactly on the opening like a glove, it will generate a click electrically. Okay, eureka, like that. So yes, the taste is detected. Different tastes are detected by different sensors. Similarly, different smells are detected by different smell sensors on the upper part of buccal cavity, that is upper surface of mouth. So smell sensors sense the fitting of shape and size of certain aromatic molecules and which fit into cavities designed to detect them as extension of sensors at cell surface. So they give electrical input to nerve upon successful detection. And ionic sensing happens at the tongue for the taste part. So taste happens at ionic level. And aromatic molecules are sensed by the shape and size, by the smell sensors. And touch, pressure, and pain and heat sensors are supposed to be embedded in skin and muscles at several places, and they are mapped location wise. The body knows which sensor is signaling from where, which part of the body. So they are mapped. Okay. And in the skin, there are hair like extension probes, which extend the sensitivity of touch and presence of static charge on hair. And then there are erector muscles, which can make the hair stand literally. Yeah. And sound sensing is done by the inner ear, of course, and there is a stereo sense which is associated with by post processing of the two signals of left and right ear. So to incorporate the element of depth. So also ears have those three horns which are sensing the stability of the head, the gyro stability of the head, so to say. And eyes have a mapped array of rod and cone cells in retina, and they also have stereo sense through post processing, and they have tracking and focusing function to aid it further. This is typically a skin with several kind of sensors seen here. In the folds closely to the surface, upper surface, are the touch sensors. Deep inside, like this bulb, these are the pressure sensors, and also heat sensors are there. Okay, so there are several layers of dermal cells, the layered cells that one can see, and then hair follicles are also present with sweet gland and sebaceous gland, that is oil gland, and hair stacks are surrounded with nerves, as you can see here. If the hair moves, it can be sensed here, right? And touch sensors are right here, and pressure sensors are somewhere. So that's all for the day. So we have seen today a lot of variety of cells and tissues diversifying from a single egg cell fusion of two sites, the ova and sperm, and how the genetic database gets modified, how it translates into so many interesting different properties, different specializations that make our body tissues. Okay, bye bye for the day and see you for the next talk. In the next talk, we'll be talking about the energy house of the cell, that is mitochondria, how the body manages the fire. Okay, because oxygen is there, fuel is there, which can lead to fire everywhere. So, how does the body prevent fire and how does the body tame fire? That will be the next talk. So, tune in for the next talk. Until then, bye bye. See you.